Okay, so the final speaker is Charlie Baer from University of Florida.
All right, thanks for the chance to be up here and to hold forth in this great meeting. So this work was done by the first four people here, actually by Sarah and Armand, and I'm just kind of carrying the ball for the last 1 yard here. But so if you're drinking in the bar, talking about what is life, these are kind of the first things you come up with, right? Well, it's got to replicate, and it's got to metabolize, and for it to get anywhere, there has to be some sort of association between the two. So a properly functioning organism needs to maintain metabolic homeostasis, right? We know that. Now here's kind of where I come in. So deleterious mutations, by definition, impair the function of the organism somehow. So ergo, it stands to reason that deleterious mutations must affect the metabolism of the organism somehow. So here's what I'm going to talk about. So what are the cumulative effects of spontaneous mutations, most of or many of which are deleterious, on the metabolome of C. elegans? And that's pretty light there. This is a sort of artist's depiction of the metabolome. In reality, of course, it's way more complicated than that. So possibly there are one or two people in the room here who don't know the mechanics of a mutation accumulation experiment. And, there we go. So I will refresh everybody's memory. So to do a MA experiment, mutation accumulation, you start out with an ancestral population that is highly genetically homogeneous. And you can do that easily in worms or in C. elegans because they're selfers. So we self them into near homozygosity, and then we grow up a bunch of them. We replicate that population many times. We did this 100 times. And then you propagate each of these replicate populations independently over many generations at very small population size. And since these are worms or self-fertile hermaphrodites, we can pick a single immature hermaphrodite every generation, propagate it. So over the course of evolution, for each of these replicate MA lines, the effective population size is just about 1, right? So that's about as inefficient as selection can get. So all but the most highly deleterious mutations will accumulate as if they are selectively neutral in this case. So, and the especially cool thing about worms is, so this represents our ancestral line. We let them evolve for 250 generations or so. The worms you can put in the freezer and resuscitate, and then you can compare the set of mutation accumulation lines, each of which has accumulated, presumably, its own unique set of mutations to their common unmutated ancestor, and we've done that. So this is sort of the first principle of mutation accumulation. On average, relative fitness will decline. Sometimes it doesn't, but almost always it does. So here I'm sort of plotting relative fitness against generations of mutation accumulation. That's what you see. So you can look at various traits that of the organism that you might be interested, and they might do different things. And for example, if you have a trait that we know is sort of positively associated with fitness, like body size typically, body size tends to decline with mutation accumulation. We call this sort of the change in the mean as I'm calling the mutational bias, and I represent that by delta M, so just change in the mean. You can have another trait, in this case time to maturity, which we think, sort of the longer it takes you to develop, the lower your fitness. A trait like that might increase with mutation accumulation. So the other sort of bit of information you can get out of mutation accumulation data is the change in the genetic variant, the change in the among line variance. So if you look at the variation among those mutation accumulation lines, it starts out at 0, right, because the lines were genetically homogeneous when you started, and as mutation accumulation proceeds, the among line variance increases, and the rate of increase of the among line variance, that's the increase in genetic variance, presumably, and we call that the mutational variance. So you can sort of parse these two composite statistics down into their components. So the change in the mean is just equal to the genome-wide mutation rate, or how many mutations you have, times their average effect. And each of those are sort of, this is summed over all loci, mutation rates at all loci, and this is the average effects over all loci. And the variance you can further break down into the mutation rate times the squared effect of the mutations. So here's what we did. So here's my MA experiment. We started out with 100. Actually, I started this out many years ago in Mike's lab, and I'm still mining the data off him. But so for what I'm going to report here, we had 43 MA lines that were derived from the N2 strain, and N2 is just sort of the white lab mouse of C. elegans. It's highly atypical. So we accumulated mutations for about 250 generations of single hermaphrodite descent. So the effective size is approximately 1. So in the experiment that Armand and Jake and Sarah did, so we were shooting for 5 replicates per line. Not all of them worked, so we got about 4 replicates per line. The common ancestor, this guy, was thought out, and they got 9 replicates of that one. Probably they were shooting for 10 and 1 didn't work or something. So then, and I know basically nothing about actual metabolomics, but Jake does. So they extracted the metabolites using standard methods, and they quantified the metabolites by GC mass spec. And so this will be coming out in Evolution shortly, so you can sort of read up on what they did. So here are the data. So you guys in the back, of course, can't read these, but there's 29 metabolites here. So 2-hydroxyproline, adenosine, alanine, et cetera, all the way down to xanthine. And so what you can see is sort of the majority of them decline. So this is the change in the mean. This is delta M. So if there's no change, it's 0. So most of them declined, but some of them increased, and some of them increased by a lot. And the orange line here, this is sort of a reference line. This is the average rate of decay of fitness, or of lifetime reproduction, which is a reasonable estimate of fitness. And I'm really, really confident in this number. That's about 0.1% per generation. That's how fast N2 declines in fitness with MA. So most of these metabolites are kind of doing like that, but a few of them, and sort of so you look at the scale, that's 0.6 on the scale, and so 4, but this would be like up at the roof of the building, right? 4 of those really, really increased. Now, there's more to it. There's a little more to it than that. So these are the 4 metabolites for which they had those really large increases: nicotinic acid, succinic acid, xanthine, and adenosine. So the orange represents, that's the mean of those 9 replicates of the ancestor. And these are the means of all the MA lines. And so if the ancestor is very near 0, then as the mean increases, the relative percent increase is really going to shoot up because you start out very small. Now, in principle, we could have seen a big decrease in metabolite concentration, but we never did. Okay, so here is this is the mutational variance, the squared CV. Again, sort of over all 29 metabolites. The blue line, again, this represents the average mutational variance for lifetime reproduction. I'm reasonably confident in that too. And again, there was, a lot of them are kind of on average there, and then some of them are substantially larger. right? And so again, this is not scaled to the control mean. This is scaled to the MA mean. So this is real variance here. So the last bit is what about the relationships between these metabolites? We got 29 metabolites, so we did a principal component analysis. On the left, I'm just sort of plotting out the eigenvalues against PC whatever, right? So here's the eigenvalues just, and here is the fraction of the variance explained by the PC. So the first 8 PCs explain about 80% of the variance. So, what is the underlying, so that's phenotypic variance. What is the underlying genetic architecture of the principal components of the metabolites? So here's the first 8 PCs. This is the fraction of the variance explained, right? So PC1 explains about a quarter of the variance, PC8, and these guys explain about 2.5% of the variance. This is the mutational heritability. So the mutational heritability is the among line variance, or is basically the mutational variance scaled by the environmental variance. And this line I have even more confidence in. That is mutational heritability of 0.001, and that is kind of the canonical value for pretty much anything ever except maybe transcript abundance. So PC1 is absolutely not heritable. So that's, presumably that's the variation due to random experimental error and noise. The remaining 8 PCs are all significantly heritable, so there's significant mutational variance for these. Actually, let me go back here. So this sort of the non-uniformity of the eigenvalues there, that tells you there are genetic or phenotypic correlations at any rate. And this tells you that there is substantial independence in the data even though there are those phenotypic correlations because the PCs themselves are uncorrelated. So I will conclude. So first of all, and this, as far as I know, this is the first time anybody has done this kind of experiment. The metabolome, or at least as represented by these 29 metabolites, presents a non-trivial mutational target. So this is both in terms of the mutational variance and the mutational heritability of the PCs. Metabolites that have large mutational biases increase in concentration. So presumably, the well-functioning worm does not have very much of these guys, and as they become mutationally degraded, they increase in concentration. We didn't see any for which there was a concomitant large decrease in concentration. And finally, there would appear to be, relatively many independent possible avenues for evolution of the metabolome, given the significant mutational heritabilities of the PCs. So quickly, where I want to go in the future, we want to sort of formally construct the metabolic network. Recently, there was one published for C. elegans, so we're optimistic this will come to fruition. Interested in it. So we looked at correlations between the metabolite concentrations and fitness. Interestingly, those 4 that changed a lot were not correlated with fitness, but we don't have very good estimates of fitness for these guys. And finally, I would very much like to compare the mutational variance to the standing genetic variance to infer something about selection. And thanks to those fine people and funding, and I'll take questions.
I was wondering if you can speculate a little bit on what molecular processes happen to increase this amount of metabolites, like the ones that increase largely. Do you think it's maybe like an enzyme in the middle of a pathway that gets hit by the mutation and then some intermediary products accumulates and can be degraded, something like that?
So that's what I guess you could call an inborn error of metabolism. That is sort of very analogous to like human genetic disease, metabolic diseases. I mean, so I don't, we don't know, but that is at least consistent with breaking an enzyme somewhere in a pathway and seeing its the precursor just sort of cool up. Yeah, Eric.
Sort of on the same lines, I noticed that two of the big ones were adenosine and ribose, which are both components of ATP, right? So could it be that partly that's elevated because they're just other systems that use ATP are broken and they're just kind of wallowing in excess ATP or its components?
That's as good as anything I can think of, but yeah, so I mean, it will be very interesting to do this again and see if you see the same kinds of things. Yes, sir.
What's the minimal mutation for early maturity? For example, from 5 mode to reduce to into 4 or into 3.
I'm sorry. If you could step away from the microphone.
Yeah. What's the minimal mutation for early maturity?
Oh, the minimal mutation for early maturity?
Yes.
I've never looked at timing of maturity. I don't know. Is the short answer. I would suspect it's on the order of lifetime reproductive success and not very different from that, but I'm kind of guessing.
For nematode, from larvae Y to larvae 2 to larvae 3, so the larvae 4, the adult.
So in terms
so
of
can we have reduced Y larval stage by mutation?
So I've never done that experiment, so I can't tell you.
Thank you.
Sorry. Yeah, Art.
So just nice work, Charlie. And I just wanted to take this opportunity to plug a workshop tomorrow on model organism metabolomics that is related to this. You've sort of done founding work in this area. So anybody interested, 8 a.m. tomorrow, Laura Reed's organizing.